It's a pleasure to introduce today's speaker, Dr. Graham Coop, who's our neighbor at UC Davis. So he's just down the road. Graham, I first came to know of Graham when he was a postdoc at the University of Chicago working with Jonathan Pritchard. He also did a lot of work with Molly Przeworski and Anna DiRienzo there. He's a theoretical population geneticist who's developed some beautiful methods for inferring alleles that are under selection in natural populations. I guess that's the most general way of saying this. He's been interested in a wide variety of fundamental topics in population genetics, including detecting selection and recombination, inferences about recombination in natural populations. He received his DPhil, which is a PhD in English. What's only in Cambridge?
Oxford and Cambridge.
Oxford and Cambridge.
Everywhere else PhD.
All right, so he, he has that accent that, to our ears makes everyone sound very smart too. You can judge for yourself. Anyway, he got his DPhil in statistics with Griffiths at Oxford University, and then came to the University of Chicago in 2004, and was there from 2004 to 2008. He joined the faculty at Davis in 2008, and he's now an associate professor there. And today he's going to be speaking about the geographic signal of polygenic adaptation in humans. And I should say that many of the methods that he's developed and were first used in humans are very widely applicable, and we'll probably get a sense of that today. But certainly people in my lab are very actively using these methods. So I think there's a lot of potential for extending this to non-model organisms. So welcome, Graham.
Okay, thanks, Michael. And I should say I'm also going to be a long-term visitor at the Simons Institute, so if people want to talk to me, I'll be generally around this month. So as Michael says, I'm faculty in the Evolution and Ecology Department at UC Davis and the Center for Population Biology. And in general, in my group, we try and think about population genetics and how we can learn about the evolutionary process using population genetic data. And in today's talk, I'm going to talk to you about some work we've been doing on thinking about detecting selection when selection acts on polygenic traits. And first of all, I should say that this work has all been done by a great student in my lab, Jeremy Berg, who sat back there. And so Jeremy and I have been working on methods to think about selection acting on polygenic traits. And this work is all available as a preprint paper and is available on the archive, the preprint archive, and we've also written some blog posts about it at Haldane Civ. And hopefully you guys in the back of the room can see these things, okay? Hopefully the font will usually be slightly bigger than that. Okay, so as population geneticists and evolutionary geneticists, there's two broad sets of approaches we take to study selection in natural populations. The first is a quantitative genetic approach, where we take a phenotype first based approach and study natural variation in adaptive phenotypes, and look at the measures such as heritability, and look at the short-term turnover in natural phenotypes within natural populations over relatively short time spans potentially. And these approaches are really great because they allow us to study natural selection in the fields with relatively little knowledge of the genetic basis of those phenotypes. And those approaches have been really quite successful at understanding selection in the field. So that's the quantitative genetic approach often to studying natural selection. There's also the population genetic approach that tends to take a genes first approach, where we study genetic variation genome-wide, and we can use that genetic variation, as you know, to study patterns of population history, and also try to identify the loci underlying adaptive traits in a relatively unbiased way by looking for certain characteristic signals of natural selection having acted in long term to generate allele frequency differences between populations. So that's based on these sorts of simple cartoon models we have of how adaptation occurs in natural populations. So as first introduced by Maynard Smith and Haig back in the 70s, a lot of our intuition about how we can detect selection acting on particular phenotypes from a population genetic way of thinking about the world is through this full sweeps model, where we imagine a set of haplotypes segregating in the population, and we're looking across the genomes of a number of individuals. And before selection acts, there's some amount of standing genetic variation for that phenotype in the population. The selected mutation arises on a particular background, increases in frequency rapidly within the population, dragging with it the haplotype on which it arose through the hitchhiking effect, right? So the selected mutation reaches high frequency, and there are various other mutations which happen to occur on the same genetic background as that selected allele, which have also increased in frequency within the population. So we can try and detect selection acting on new mutations which have rapidly increased in frequency in the population. For example, by looking for large allele frequency differences between populations. So I'm showing you one particularly nice example here of looking at the gene SLC24A5, which is one of the largest allele frequency differences between different human populations. And this allele is at high frequency across all of Western Eurasia and almost absent in East Asia, Africa, and the Americas. And this allele at SLC24A5 is one of the alleles associated with a lightening of skin pigmentation in Europeans and other Western Eurasians. So that allele has gone to very high frequency very rapidly in those populations, and in doing so, it's dragged with it the haplotype on which it arose. It's wiped out genetic diversity in a broad swathe of the genome around it. So if we look at levels of heterozygosity, a measure of genetic diversity, in the regions surrounding this mutation, we see that there's, in general, relatively high levels for humans of levels of genetic diversity in the region outside that region, so away from that gene. But in the region around that gene, we see a large dip in heterozygosity, indicating that we've erased genetic diversity as this mutation swept to fixation. Similarly, we can look at patterns of haplotype variation around this mutation. And so if we look in African populations, East Asian populations, the Americas or Oceania, we see that there's a relative jumble of haplotypes looking across the genome and across individuals, colouring haplotypes by their similarity to each other here. But in these populations where this allele's recently gone to fixation, we see that there's a dominant haplotype at high frequency in those populations. And that's the haplotype which that selected mutation arose on. That's the haplotype that's hitchhiked to fixation with that selected allele. So as human population geneticists, as population geneticists, we've been looking for these types of signals in a variety of populations and in a variety of genome-wide data sets, looking to see these signals of high FST alleles between populations, reduced genetic diversity, and extended haplotypes associated with the selected allele. And in humans, we found a number of nice examples, some very large outliers in the genome, that seem potentially involved in human adaptation as populations have moved around the world. But also we've started to realise that actually there's relatively fewer of these signals segregating in the genome than we might have previously thought. So we can try and go out and look for general signals of this type of adaptation occurring in aggregate across many sites in order to try and see if we have a genome-wide signal of adaptation having occurred through this simple cartoon model of this full sweep. So for example, what I'm showing you here is some data taken from a paper we had back in 2009. And what we're doing here is we're taking the top 50 high FST SNPs between East Asia and Africa. So the most extreme sites in the genome corresponding to high FST between East Asia and Africa. And we're looking at a measure of haplotype homozygosity, looking at a measure of how much of a reduction in genetic diversity we see around these high FST SNPs. So we're plotting this measure, XBEHH, which is a measure of the reduction in haplotype heterozygosity within a population, that reduction in genetic diversity within a population, for these high FST SNPs in East Asians. So we can see that that distribution of those high FST SNPs is shifted relative to what we expect sampling random SNPs in the genome. So we can see that these high FST SNPs, on average, have greater reduction in haplotype heterozygosity around them, suggesting that some of them have indeed potentially swept to fixation. But we can also do simulations of what we'd expect under strong selection acting on these mutations from the moment they enter the population. So here I'm showing you the distribution of the haplotype homozygosity under some simulations where selection acts on the mutation from the moment it enters the population and sweeps it up to fixation. And there we can see that there's a stronger shift away from that neutral distribution, from that genome-wide distribution. So what we're seeing is, when we look at the sites in the genome, these high FST SNPs, the top 50 SNPs in the genome, between East Asia, that differentiate East Asia and Africans, there's some reduction in haplotype homozygosity surrounding those SNPs, but it's potentially nowhere near as strong as we'd expect under strong simulations of selection. There are many high FST SNPs which seem to have relatively little reduction in haplotype heterozygosity around them. So this is potentially a signal that some of these high FST SNPs, which have been driven through populations, potentially were standing variation when they entered the population, rather than being selected on from the moment they entered the population. Another signal that just has come out in the past few years that started to indicate that there may be relatively few hard sweeps, these sweeps from a new mutation entering the population, is this paper by Ryan Hernandez et al., which appeared just a couple of years ago. And what you're looking at here is you're looking at a signal aggregated over many sites in the genome. And you're looking at a measure of genetic diversity again. You could think about this as being heterozygosity. And you're looking around a particular substitution, and this has been averaged over every single substitution on the human lineage. And you're looking at the scale of diversity, on average, around non-synonymous substitutions. So non-synonymous substitutions on the human lineage are potentially our best guess at the types of adaptive alleles which maybe have been swept through the population. They putatively have some function as they're non-synonymous. When we look around those alleles, we see that there is a reduction in heterozygosity, in genetic diversity, close to those sites, those non-synonymous alleles have gone to fixation. So that's potentially a signal of selection, right? We're seeing, on average, that when we look around a non-synonymous substitution, we're seeing a dip in genetic diversity, characteristic of what Maynard, Smith and Haig told us to expect around where a selected mutation had gone to fixation. However, what Hernandez et al. Also did was they also did this around synonymous site substitutions. And you're seeing here the synonymous sites diversity around a synonymous substitution, also plotted out, aggregated over all such substitutions in the genome. So you can see that when they look around synonymous substitutions, they also see almost the same dip in heterozygosity as they saw around an amino acid substitution. So they took this as evidence that relatively few of those non-synonymous substitutions have been driven by strong selection. In fact, they can estimate that relatively few of them have been by doing some simple simulations on top of this. And in fact, this reduction in genetic diversity, which we see both around synonymous substitutions and non-synonymous substitutions, is potentially due to some other form of link selection other than full sweeps, such as background selection. So these pieces of evidence and a number of other pieces of evidence to us suggest that there's relatively few full sweeps to be found in the human genome. There are relatively few sites where a new mutation has rapidly increased in frequency, dragging with it the haplotype on which it arose due to selection. Rather, we think that humans have clearly adapted to their environments, right, as all populations have. But maybe it doesn't occur through this simple cartoon model where a new mutation increases in frequency from the moment it enters the population. Rather, we've known for a long time that adaptation may often occur through selection on standing variation. There's plenty of standing variation for a range of phenotypes in the population. And as population geneticists, we're now going to have to start to get to grips with looking for signals of selection on standing variation. In particular, we might imagine that when a selected mutation enters the population, it may not originally be adaptive. It may segregate at low frequency due to the fact that it's a mutation selection balance, or it's relatively neutral in the environment it's present in, such that a mutation could be found on a range of different haplotypes within the population. So that when selection pressures switch, allowing that allele to increase in frequency in the population, rather than taking with it a single haplotype to fixation, it may take with it a number of haplotypes on which it was present in the population as standing variation. So this acts to much diminish our signal of selection having acted on this allele. This allele's still gone to high frequency in the population, but it hasn't taken with it a large haplotype on which it arose. It's taken a number of haplotypes of intermediate frequency in the population. So we and others are working on methods to detect selection on standing variation, and I'll be happy to talk to you about that at some other time. But we also know that when selection acts on natural variation, it may be acting on many mutations in concert in the genome. So that we might imagine there's actually a set of loci where adaptive mutations are presently segregating in the population, such that if selection pressures change, these alleles may all increase in frequency in concert within the population, right? In that case, there may be no strong signal in terms of allele frequency differences between the original population and now the adaptive population. There may also be no strong signal in patterns of haplotypes within those populations. And indeed, this is typically the model that quantitative geneticists are thinking about, right? As quantitative geneticists, the reason we can ignore the genetic basis of our traits is we tend to think that there are many loci segregating variation in the genome which controls our phenotype. And when selection acts on those phenotypes, adaptation occurs through small allele frequency shifts at many different loci, leading to rapid adaptation, but no strong signal of FST at any of those loci, right? So there's a difference in how quantitative geneticists and population geneticists think about looking for selection. But there's now an increasing opportunity for us to try and start to combine those techniques for looking at selection. And that's because over the past few years, people have started to do genome-wide association studies to try and map the genetic basis of of a range of different phenotypes, including potentially adaptive phenotypes, within human populations. So people have been going out, and as you probably know, genotyping tens of thousands of individuals, phenotyping those individuals as well, and looking for genotype by phenotype associations to identify the genetic basis of a range of different phenotypes, often disease phenotypes, but also other phenotypes within natural populations. So these efforts are carried out in human populations, and in general have started to identify some of the loci underpinning natural phenotypic variation in human populations. So typically, those genome-wide association studies have confirmed that polygenic view of phenotypic variation generally, these studies, rather than identifying large effect mutations at a small number of loci, identify that the genetic basis of a range of different phenotypes is due to tens or hundreds of loci, all segregating in natural populations. So for example, here you're looking at a study for body mass index, and you're looking at the p-values across the genome at a large number of SNPs, and they've identified a large number of associations between genotype and phenotype. Each one of these alleles has a very small, seemingly additive effect on the phenotype of interest. And these loci explain only a relatively small proportion of the phenotypic variation, potentially because there's a larger number of loci yet to be discovered. Okay, so there are other genome-wide association studies. For example, here's a genome-wide association study on skin and eye pigmentation in an African American populations. And in some cases, these studies are finding large effect alleles. For example, the alleles associated with skin pigmentation, that seems to be a relatively simpler phenotype genetically. So to date, there's a small number of loci that have been mapped associated with natural skin pigmentation variation in humans. And you're seeing once again this same chart looking at the loci they've identified. So human geneticists are doing many of these association studies to identify the genetic basis of a range of different phenotypes. These are also starting to be conducted in a range of other populations, such as in Arabidopsis thaliana or in Drosophila. And in general, these studies are also identifying large numbers of loci associated with a range of ecologically important traits. So for example, this is data from that Arabidopsis study, and they've looked at a large number of phenotypes in a set of natural isolates taken from across the range of Arabidopsis thaliana. And looking at a range of different phenotypes, they've identified many different loci influencing a range of different phenotypes. Once again confirming the idea that the majority of the genetic basis of phenotypic variation is potentially due to this sort of polygenic number of loci. There are some large effect alleles associated with phenotypes, but there are also a large number of loci which contribute to any phenotypic variation in population. So the question is, okay, we're now starting to learn about the genetic basis of a range of different phenotypes. That's great, right? That's what we as population geneticists, as quantitative geneticists, are interested in. We can now start to use these genome-wide association studies to tell us about the loci associated with a particular phenotype. And so if we want to know how selection has acted on a particular phenotype, we can look for signals of selection at these loci associated with the phenotype. So as population geneticists, we and others have been taking these genome-wide association study loci and looking to see, for example, are they outliers in terms of allele frequency differentiation, in terms of FST, for example. And those have identified that some phenotypes do seem to have larger amounts of differentiation at those underlying loci than you'd expect given the rest of the genome-wide background. But those types of approaches of using population genetic methods to look at these quantitative trait loci are implicitly usually assuming that same model where selection acts on a mutation from the moment it arrives in the population, looking for these sort of population genetic signals of long haplotypes, reduced diversity, very large allele frequency differences between populations. And yet we know that given that there are tens or hundreds of loci involved in the genetic basis of these traits, we don't expect to see large allele frequency differences at these loci, even if selection has acted quite strongly on these traits. Rather, we'd expect that selection may have driven driven phenotypic differentiation across populations, but it may have done so by relatively small allele frequency changes at many loci. So, what Jeremy and I have been trying to do, and a number of other people have been doing as well, is trying to return to sort of quantitative genetic ideas, but now using the fact that we know some of the loci involved in the phenotype of interest. What we're doing is we're trying to use these genome-wide association studies to try and construct genetic additive values for populations. So what do I mean by that? Well, somebody has very kindly performed a genome-wide association study for some phenotype of interest in a particular population. They've estimated, well, they found L loci associated with a particular phenotype. So these are L loci which are significantly associated between genotype and phenotype. And they've estimated, usually, additive effect sizes for those L loci. So this is the additive effect. This is the fact that having an extra copy of this allele at this locus increases your phenotype by alpha 1 at the first locus, alpha L at the L locus, right? That's what we estimate when we do associations between genotype and phenotype, are these additive effect sizes. Okay, well, we're going to do the silly thing of just taking these at face value and constructing phenotypes in other populations given this additive model of how these loci act. In particular, we're going to take some other population, M, and we're going to take the set of allele frequencies in that population M at our first locus all the way through to our L locus. And we can try and construct an additive genetic value for our phenotype, for this population, given these effect sizes and given these allele frequencies, simply by saying that the mean additive value for the phenotype in this population M is found by just summing up the additive effect at each one of those underlying loci, weighted by the allele frequency in that population. And there's a factor of 2 there because as humans we're diploids, right? So this is the additive genetic value for the population M using these L loci and their effect sizes. Okay. So we might like to think of those additive genetic values as a phenotype for the population, right? But they're obviously not predictive phenotypes for the population because we know that we've just assumed additivity. We're assuming that there's no dominance or epistasis, that these additive effect sizes for these alleles hold true in these other populations. So we've just completely ignored this whole issue of dominance and epistasis. So that might lead to our predictive phenotypes not being good estimates of the phenotype in the population. Also, these genome-wide association studies that people are conducting usually don't identify the causal mutation. They just find something that's genetically associated with it, i.e. is in linkage equilibrium with it. So we often don't know the causal mutation, but simply something correlated with it. We also know that we're ignoring all of the interesting gene-by-environment interactions, right? In using these additive effect sizes, estimated in one population, and using those in a different population, we're completely ignoring the issue of gene-by-environment interactions. Also, these genome-wide association studies are potentially only identifying the genetic underpinnings of about 15% of the variants that's segregating the population. So they're missing a large number of other variants underpinning the variation in that phenotype. Despite that, these alleles which have been identified to date do represent a rich source of information about phenotypic variation across populations, even though we shouldn't think of these as predicted phenotypes for these populations. The really good news for us, and why we're thinking this is not such a stupid thing to do, is we've actually constructed a perfectly additive phenotype for the population, a perfectly additive genetic value for the population, using these loci. So, why that's nice is we can actually use these additive values that we've predicted for populations to look for selection acting in aggregate on all of the loci underpinning these additive genetic values. And all of these assumptions that we've sort of violated in constructing these genetic values for these populations are going to make it difficult to treat our predicted genetic values for populations as predicted phenotypes. But none of these assumptions are going to violate the test of selection that we're going to set up. Okay. So as an example of doing this, we're going to take the 180 loci to date associated with human height. And that's good because height is this classic polygenic phenotype in populations. The body size is a classic polygenic phenotype in many populations. And it's also nice because other investigators have also identified a signal of selection acting on these loci in aggregates. So we're going to use this as a sort of case study to work through this method to look to see whether we can see selection acting on human height across populations simply by using the genetic data alone. Okay, so what you're looking at here is this constructed additive value for height across 52 human populations that consist of the Human Genome Diversity Panel data. So from Europe, the Middle East, Central Asia, East Asia, Oceania, the Americas, and Africa. And so we're looking at this additive genetic value constructed using those 180 loci for height across these 52 populations. So you can see that that constructed phenotype varies across populations, right? We can see that there's differences between different continental regions of these human populations. And that could potentially be due to the fact that selection has acted on these loci, leading to allele frequency changes between these populations, right? But it's also the case that this simply could be due to genetic drift, right? This could be the case that these loci underlying our phenotype, height, differ in frequency across populations simply due to genetic drift. And so we might expect to see this variation across populations simply by genetic drift, and we'd certainly expect to see that different continental regions will tend to cluster in their phenotype, potentially due to selection, but also simply due to the fact that there's shared evolutionary history there, shared genetic drift, so that the loci may have simply drifted up on average in frequency in Europe and potentially drifted down in frequency in Africa. But the nice thing is, we know the loci underlying these constructed phenotypes, right? We've simply added them up together. So for example, here's one of the loci associated with height across these populations. And I'm showing you the allele frequency variation across those populations. And this locus differs in allele frequency across these populations, potentially due to selection, but potentially more likely just simply due to genetic drift. So we're going to need a null model to describe the amount of variation we see in this constructed phenotype across populations. So we're going to construct a simple null model to explain whether this is likely to be seen under genetic drift. And this is nicely going to build back into some old quantitative genetic technology. Okay, so what's our null model? We're going to assume that there's some ancestral frequency at a locus epsilon, and there's been some amount of genetic drift since that ancestral population. So our allele frequency within the population is now P. So you might imagine, for example, that our ancestral allele frequency was a half, and you're looking at the distribution of P following a number of generations of genetic drift since that ancestral population. So that now P is centered around that a half frequency, but there's some amount of genetic drift there. Okay, so we're going to say that allele frequencies are normally distributed around this ancestral mean epsilon, and there's going to be a variance in allele frequencies, epsilon 1 minus epsilon, acknowledging the fact that genetic drift is binomial sampling compounded across the generations, and this parameter f, which simply represents the number of generations since that ancestral population, scaled by the effective population size. So it's the amount of genetic drift you could think about it as being like FST. Okay, so in reality we don't just have one population, we have a number of populations. And so we're interested in the allele frequencies across these populations. Those allele frequencies have drifted away from this ancestral allele frequency. So they're variable around that ancestral frequency. They co-vary with each other due to the fact that populations have differing amounts of shared history, differing amounts of shared drift. So for example, in this situation here, you could imagine that these two populations will be more similar in allele frequency due to the fact they share genetic drift along this lineage here, right? Okay, so we're going to assume that once again, allele frequencies, well, they're going to be centered on that ancestral frequency. But we're going to say that they're, once again, normally distributed. But we're going to say that there's now a multivariate normal distribution, where the variance, once again, has this binomial sampling term. And now there's going to be a matrix of covariance in allele frequencies between populations. So that's going to say to the extent to which populations covary in the deviation they take from this ancestral mean, due to the fact that populations have differing amounts of shared history. Okay, so we've said that our allele frequency is multivariate normal around some ancestral mean with some covariance matrix F. Okay, well, in constructing these genetic values for these populations, what we've done is we've simply taken a weighted sum of allele frequencies to construct these genetic values for these populations. So that means that our phenotypes are also multivariate normal. And they're multivariate normal around some ancestral mean with some covariance matrix, which is the original covariance matrix of these allele frequencies, now weighted by this term. And that term is simply the additive genetic variance in this ancestral population. Okay, so we now have a model to describe allele frequency variance around the population. So it turns out that we can quite nicely standardize these allele frequencies. So we have a set of phenotypes distributed around some global mean with some covariance matrix. We can write down a standardized set of phenotypes, removing the effect of this covariance matrix. And those standardized set of phenotypes are now centered around 0 because we've minus off the mean. And we've completely removed the effect of the covariance due to that shared genetic drift. Okay. So, given that, a natural measure of the variation in the phenotype we see is simply the variance of the phenotype across populations, accounting for this covariance matrix. So we can write this statistic that we're going to call QX, which is the variance in our standardized phenotype. So you can think about this as just the variance of the phenotype, having removed the effect of shared population history. And this is a good statistic to look for adaptation having acted on this phenotype differentially across populations. Because if selection's acted on our phenotype differentially across populations, that's going to lead to our phenotype being too variable across populations compared to our null model of just simply genetic drift. What's nice is this statistic QX that we've set up is actually chi-squared distributed with the number of populations minus 1 degrees of freedom. Okay. So we were quite pleased with ourselves having come up with this way of looking for selection on these phenotypes, these purely genetic phenotypes. But we also realized that what we'd actually done along the way was partially reinvent this statistic called QST. So turns out quantitative geneticists really knew what they were doing when they were looking for selection on phenotypes, unsurprisingly. They proposed this statistic QST, which looked at the variance in phenotypes, genetic phenotypes calculated from crosses across populations, and compared it to FST. And if there was no hidden population structure, if all populations were equally related to each other, but just drifted away from each other, they knew that QST divided by FST was actually chi-square distributed. And this test has been used for a long time to look for selection acting on phenotypes in natural populations. So what we've done is we'd reinvented QST, but we've done so in such a way that actually accounts for population structure. So populations can be differentially related to each other, and we now have a way to remove the effect of shared genetic drift to allow us to look for selection acting on phenotypes, accounting for differential relatedness across populations. Okay, so what's nice is that because we've genetically constructed a phenotype, this phenotype is guaranteed to be perfectly additive. And so all of the assumptions about QST that people have worried about over the years actually are completely satisfied by our model because we have purely additive phenotypes. Okay, so this is this QX statistic, this standardized variance for human height in the population. And we can compare that to this chi-square distribution that we'd expect under this null model of genetic drift. So human height across our 52 populations is indeed more variable across populations than we'd expect under the theoretical model of genetic drift. But you might worry about the fact that this model that we've constructed may not be particularly realistic. It may not be that this theoretical distribution closely matches what you'd predict under a model of genetic drift, given the data. But what's nice is now that we have large amounts of data, we can actually go out and we can numerically construct this null distribution simply by reassigning those 180 effect sizes for our height loci to other loci at random in the genome, matching them in all sorts of ways to make sure that they're a realistic null model for those SNPs. And when we do that, we find that our null prediction of FST from theory and from this empirical null constructed by reassigning effect sizes of the loci really quite nicely match each other. And according to both distributions, height, human height, looks like an outlier across populations. Okay. What's also nice is we can actually decompose this QS statistic, and it turns out that quantitative geneticists have known this for some time as well, into an FST-like component and an LD-like component. So, what you're looking at here is this FST component for height. So this is looking on average, are our loci overly differentiated on average across populations? And we're comparing that to an empirical null distribution. And unsurprisingly, these height loci aren't overly differentiated on average. They look, on average, relatively neutral in how they vary across populations. Where our signal is coming from is due to the fact that individual alleles are co-varying across populations in a manner which is predicted by their effect sizes. So we're generating high heights in some populations, not by one locus controlling height going to high frequency, but by a coordinated shift across many loci in alleles which increase height, going up or down in frequency in concert together. That generates this covariance across loci in the deviation they take around this global mean. And that's what's driving our signal of selection on height. Not large allele frequency changes at individual loci, but rather this concerted shift at many loci across the genome. So we've been doing that for a range of other human phenotypes. So here's height, here's skin pigmentation. Skin pigmentation once again showing a strong signal of selection. We also potentially see, well, not very strongly, but a signal of selection on body mass index. But many other disease phenotypes show no signal of selection as mapped to date across these human populations. Interestingly, type 2 diabetes, which many people associate with potentially having adaptive differences between populations under this so-called thrifty gene hypothesis, doesn't seem to be strongly differentiated across populations compared to our empirical null, suggesting that incidence of type 2 diabetes, the genetic underpinnings of that as mapped to date, that hasn't been driven by selection across populations. Okay, so in the interest of time, I'll quickly move on and just talk a little bit about some of the downsides of what we're doing. So we think we have a really quite robust way to look at selection acting on phenotypes across populations that's closely related to these QST measures, but really satisfies all of the assumptions of QST because we've constructed these perfectly additive phenotypes. The disadvantage is that in constructing those additive phenotypes, given one set of additive effects estimated in a population, we're unsure about what exactly is driving the signal of selection across populations. So for example, we're returning to a height example here. Don't worry about the different sizes of the dots here. We see a strong difference in height, this constructed height, between African populations and populations outside Africa. That's a signal that selection has acted in concert on these loci underlying human heights. But we don't really know that these African populations are genetically smaller than these European populations. Almost surely they're potentially not. What could happen here, right, is that there's another set of loci in these African populations that has yet to be mapped, which leads these populations also being taller in height. So there could also be gene by environmental interactions here that we're completely ignoring, that could also explain why these different phenotypes that we're predicting are relatively poorly matching observed phenotypes. But we think that as people start conducting genome-wide association studies in a range of other human populations, we can start to map the genetic basis of these phenotypes across a range of different populations and learn in concert about how selection has shaped allele frequencies, shaped these phenotypes across populations. Okay, so given a detailed annotation of the sites underlying a phenotype, we can detect very subtle signals of differential selection having acted on these populations. We're also developing methods to look at environmental correlations with phenotypes, and also working on methods to look at multiple correlated traits. But the question is this actually a good idea? What we're doing is in effect QST. Quantitative geneticists have been estimating QST, comparing it to FST, for decades, and looking at selection acting on a variety of different phenotypes. Human geneticists have been mapping the genetic basis of these phenotypes because they're interested in the genes involved in these phenotypes. So it is a good idea to do these genome-wide association studies to map the genetic basis of phenotypes. But if we're interested in looking at selection on these phenotypes, if we were capable in humans of doing common garden studies, we'd actually be better off just doing QST than going to all of the effort that we've gone to. So, in general, quantitative genetic approaches are good ways to look at selection acting on phenotypes. As population geneticists, I think we can point to additional signals to look for selection on these phenotypes. But the ability of us to look at selection on these phenotypes is still determined by how much of the genetic variation we can explain. And it might be that using traditional quantitative genetic approaches are the right way forward. With that, I'd like to thank various people in my group and take any questions.